Let's talk about the cell cycle. It's this beautiful, super complex dance that lets every living thing on Earth grow, heal, and, make more of itself. But how does it actually work? Well, let's dive in and break it down. Just stop and think about that number for a second. 2 million every single second. It's absolutely mind-boggling. And this incredible, constant process of renewal all comes down to cell division. But here's the thing. To make a new cell, you first need a flawless copy of its instructions. So how does a cell manage to package and copy its entire genetic library without making a mistake? And that question leads us right to life's blueprint, the chromosome. See, before a cell can even think about dividing, it has this massive packaging problem to solve. How in the world do you fit 6 feet of DNA into a nucleus that's completely microscopic? I mean, without it just becoming a hopeless, tangled mess. The answer is actually a pretty brilliant system of folding and compaction. Okay, so it all starts with something called chromatin. Now, this isn't just loose DNA floating around. It's DNA that's already been wrapped around proteins to make it way more compact. You can think of it as the raw material, kind of like the thread from which the much, much denser chromosome is eventually woven. So what are these little molecular spools that the DNA wraps around? They're proteins, and they're called histones. And honestly, it's a perfect match. DNA has a negative charge, and histones have a positive one. So opposites attract. This natural pull creates a sort of electrostatic hug. And that is the very first and maybe the most crucial step in getting all that genetic material organized. So when you get a piece of DNA to wrap around a core of 8 of these histone proteins, you get a structure called a nucleosome. If you could zoom way, way in on chromatin, it would look like beads on a string, and every single one of those beads is a nucleosome. This is the next level of getting everything packed down tightly. And after all this coiling and supercoiling, and importantly, after the DNA gets copied, we finally end up with that classic shape we all recognize, the X-shaped chromosome. That X is actually made of 2 totally identical copies of the DNA, which we call sister chromatids. They're held together right in the middle at a spot called the centromere. They are perfect duplicates just waiting to be pulled apart. Okay, so now we know how the instructions are all neatly packaged. The next logical question is, how does the cell actually split them up? Well, it turns out life has come up with 2 very different strategies, and it really just depends on how complex the cell is. On one hand, you got your simple prokaryotic cells, like bacteria. They have just a single circular chromosome. Pretty easy to manage, right? But then you have complex eukaryotic cells, like ours, with a whole bunch of different linear chromosomes. You can't just split those willy-nilly. You need a much, much more precise strategy to make sure each new cell gets a full, complete set. So this diagram here shows the prokaryotic strategy. It's called binary fission, and it's really elegant in just how simple it is. As you can see, the process is incredibly direct. The cell just copies its DNA, scoots the copies apart, and then splits right down the middle. Let's just walk right through it. Step 1, replicate the chromosome. Simple enough. Step 2, separate those 2 copies to opposite ends of the cell. Step 3, build a little wall or a septum right down the middle. And finally, step 4, the cell just splits in 2. It's super fast, super efficient. Really, it's perfect for organisms that need to multiply in a big hurry. Right, but eukaryotic cells, with all their different chromosomes, they just can't afford to be that simple. It would be chaos. They need a much more tightly regulated system, something to make absolutely sure every new cell gets exactly 1 copy of each chromosome. And to do that, you need a series of what you could call molecular traffic cops. The main engines that are actually driving the cycle forward are enzymes called cyclin-dependent kinases, or CDKs for short. Now, on their own, they don't do anything. They're inactive. But when these other specific proteins called cyclins bind to them, the whole complex switches on. It starts adding phosphate groups to other proteins, which is basically like flipping a molecular switch, and that gives the go signal for the next stage of the cycle to begin. But what if something is wrong? What if the DNA got damaged? That's where checkpoints come in. Think of these as critical quality control points that can just slam on the brakes and halt the whole process. They are constantly monitoring the cell's progress, and they can totally override those go signals from the CDKs if they spot any errors. It's a system designed to prevent disaster. So, you see, the cell cycle isn't just about cranking out identical copies. No way. Different versions of this basic process are actually responsible for everything from the genetic diversity that drives evolution all the way to the process of aging itself. Take meiosis, for instance. This is a really special type of cell division that's used to create sex cells, or gametes, so things like sperm and eggs. Instead of making an identical clone, meiosis basically shuffles the genetic deck and then cuts the number of chromosomes in half. The whole point is to ensure genetic uniqueness in the offspring. And this genetic shuffling happens in two main ways. First, you've got crossing over, which is where chromosomes literally get tangled up and swap little pieces with each other. This creates brand new combinations of genes. Then you have independent assortment, which is like randomly sorting those mixed and matched chromosomes into the new cells. The end result? Every single gamete is genetically one of a kind. But the cell cycle isn't just about creating new variety, it's also about preventing dangerous errors. So what happens when a checkpoint finds damage that just can't be repaired? Well, the cell enters a state called senescence. It basically gets retired. It permanently exits the cycle and it will never, ever divide again. This is a vital safety mechanism. It's a way to stop a potentially cancerous cell from multiplying. But here's the catch. As we go through life, our cells pick up mutations, and more of them enter this retired, senescent state. And while they're not dividing anymore, they're still active. This buildup of non-dividing but still active cells is now thought to be a major reason we age, why our wounds heal slower, and why we experience other age-related declines. And that leaves us with a pretty fascinating final thought. Certain animals, like the American lobster, for example, they don't seem to experience this same kind of aging process. They just keep growing and reproducing throughout their incredibly long lives. It really makes you wonder, doesn't it? What secrets are hidden inside their cell cycle? And what could they possibly teach us about our own?